Hello and welcome to this second podcast on histone modifications. In this podcast we'll talk about histone methylation as well as some other histone modifications such as phosphorylation, ubiquitination, summylation, and ADP ribosylation. Alright, let's go ahead and get started. Okay, now let's talk about histone methylation. I introduced this topic to you a few podcasts ago in talking about the DNA methylation enzymes. But what we're specifically talking about today is this protein being methylated. And as I alluded to earlier, methylation is more complicated. It can methylate some lysines, and when it methylates those, it decreases expression. Often it does this by, it competes with acetylation. Methylation at other lysines will increase expression. So it's not as straightforward as acetylation. So like acetylation, methylation also often occurs on lysine amino acids. So let's write lysine here. And it's a positive charge. And we can add methyl groups to this, and we can add them in a slightly different way. We can either add one methyl group to it, and when we do that, we call it monomethylation. We could add two methyl groups to it, and we would call that dimethylation. We could also add a third to it, and wait for it, this will be quite shocking to you, I'm sure, but we call this trimethylation. So whether a particular lysine has 1, 2, or 3 methyl groups can determine the effect on expression it has. So this complexity is best illustrated with this example. I'm going to draw a straight piece of DNA here, and I'll draw our histone octamer here, forming the nucleosome with its tail. I'm going to draw it longer than usual here. We have these two lysines. This lysine here is on histone 3, lysine 4. This one here, also histone 3, and it's lysine 9. So they're close to each other. This histone 3, lysine 4 can be methylated. And if it's dimethylated, this gene is ready. It's not turned on yet, but there's nothing really holding it back. When it becomes trimethylated at that lysine, that means it's ready to go, and it will. Gene transcription is now active. So dimethylated, it's poised, ready to go, and then if you add a methyl group here, it allows for active transcription. So a pretty good, what we might call on-off switch. Just adding that extra methyl group makes a big difference. Now that's not the whole story, because what I just said here is only true if this lysine is acetylated. If it's acetylated, everything I just said is true. Dimethylated, it's ready. Trimethylated, it's fully expressing the gene. Now, if acetylation is blocked, a methyl group could come along here. And if it's methylated here, everything I said is wrong. This doesn't happen. It doesn't matter how many methyl groups you have attached to this lysine, transcription is blocked. So methylation offers this degree of complexity, and it makes it more difficult to understand. But you can see how these very small molecules binding to these other really small molecules can have these huge effects, and it allows for this precise regulation of genes. The precision of gene regulation has always amazed me. And then when you learn that it's just because of some methyl groups coming along here, tiny little methyl groups, kind of blows my mind, thinking everything that happens because of these tiny little molecules. It can't just be these tiny little molecules. Well, it is these tiny little molecules, but they themselves don't reduce transcription or increase transcription. Just like acetylation had the bromodomain proteins, histone methylation has their own domain. They can't be left out. And these domains are called chromodomains. So many proteins that are important in the structure of the chromatin have these chromodomains on them. Again, these are just independently folded structures on that protein. And what they do is they bind to methyls attached to lysines. Just like the bromodomains, the chromodomains can bind with rather great precision. They will bind to certain methylated lysines, but not all methylated lysine. When these chromodomains, or the proteins containing these chromodomains, bind to these methyl groups, they themselves don't directly increase or decrease expression, but rather there are other domains on that protein that can affect transcription, or these chromodomain proteins, when they bind, they recruit other enzymes into the mix. They might recruit to this region here, say, various repressors or other proteins necessary to maintain a closed chromatin state. Or down here, they might bind and they might bring in various, like your RSY SNF complexes or transcription factors. So like the bromodomains, the chromodomains serve as this anchor by binding here and recruiting other proteins that come along and do the job of decreasing or increasing gene expression. Well, I would love to talk about methylation and acetylation longer, but I think it's time for us to move on to a few other modifications. We won't say as much about these other modifications, mainly because we know a lot more about methylation and acetylation, but the literature is expanding for these others, and I want to make sure we at least mention them because we know they play an important role. Okay, so this next modification we're going to talk about is histone phosphorylation. When we talk about acetylation, the data is pretty clear that acetylation increases transcription. Phosphorylation seems to be important in other cellular processes. So what is histone phosphorylation? Well, let's draw our DNA here, and we'll draw our various histones along the DNA with their tails, their N-terminal tails. And we've talked about how they can be methylated, acetylated, but now what I want to talk about is how they're phosphorylated. So phosphorylation is the act of adding a phosphate group to an amino acid. We do this using an enzyme called a kinase. It's reversible, removing the phosphate using a phosphatase. The amino acid that this phosphate group is added to is usually a serine in the cases of histones, but it could be a tyrosine and a threonine. But one of our better examples that we know about is with serine. Oh, and I should mention that the reason it adds these phosphate groups here is each of these amino acids, serine, tyrosine, and threonines, all contain a hydroxyl group on them. And this is what the phosphate is added to. The serine that I want to talk about that gets phosphorylated is on histone 3 and serine 10. Before I talk about the role of phosphorylation of this serine, I want to take you back in time and remind you of your good friend, the cell cycle. Yes, I know you had this many years ago, and we as biologists continue to bring it back up, and so I feel obligated to bring it back up here, but I'm going to do it in the context of this problem here. To remind you, a cell enters G1 with a set of chromosomes. In S phase, that set of chromosomes is copied, and in G2 we verify, make sure there's no mutations, and this replicated chromosome now enters mitosis or meiosis, but we're going to talk about mitosis. In mitosis, it goes through prophase, metaphase, anaphase, and telophase. And then the cells separate during cytokinesis, and you end up with two cells, each with that chromosome in them. So why do I bring up the cell cycle? We notice that this serine on this histone 3 here is phosphorylated, or begins to become phosphorylated, in the prophase stage. Histone 3, S10 is phosphorylated. During metaphase, we have increased phosphorylation of this H3, S10. It peaks during metaphase, and then it gradually is dephosphorylated. And finally, once it hits this new cell stage after cytokinesis, it's no longer phosphorylated. We could draw a graph where we have prophase, metaphase, anaphase, and telophase, and over here is percent phosphorylated, and what we would see is something like this, where it begins to rise during prophase, it peaks in metaphase, and it slowly decreases in anaphase and telophase. So why is it being phosphorylated? Well, we don't think it's gene specific, because as this phosphorylation occurs, it appears to occur, and particularly as it increases, across the entire genome. All histone H3s receive this phosphorylation of serine 10. So it's not gene specific. It's likely involved in getting the chromosomes ready for proper segregation during M phase. During M phase, the chromatin has to be completely compressed into the chromosome structure. So it's possible that these phosphorylations, one thing they might do is they might prevent this tail from interacting with this tail. Because if they're not modified, unmodified tails will aggregate. Just like when you put in acetyl group here. The acetylation prevents the aggregation of these tails. Phosphorylation also has the ability to prevent the aggregation of the tails. And maybe that's important in the further compression of the chromosome structure needed for metaphase. Now, it also may play a role in transcription. Now, to talk about this role in transcription, I extended the DNA out here a little bit and drew a new histone complex here with this internal tail. And I drew it a lot longer so I could point out some of our lysines that we've talked about. Lysine 4 and lysine 9. Remember we said if lysine 9 is methylated, that results in no transcription. It blocks transcription. An interesting thing happens here is that this position 10, remember this position 10 is serine over here. These were lysines, just to remind you. If this serine is phosphorylated, a phosphate group is added to it, it blocks the methylation. Phosphorylation of this serine keeps this lysine from being methylated and would promote the acetylation of this lysine, and then in turn this gene on. So I'll say this again. Methylation of lysine 9 here is prevented if phosphorylation of serine 10 is present. The blocking of the methylation here allows the acetyl group to come in. In fact, the enzyme that comes along here and adds that acetyl group, it has a greater affinity here if this serine is phosphorylated. It could be the negative charge that hits its active site a little bit better. I'm not sure. But we do know that it prefers this lysine if serine is phosphorylated. Alright, so we talked about phosphorylation's role during mitosis, its indirect role here in transcription, and now I want to close with some observations for phosphorylation in two other areas. The first is in DNA repair. We know that this histone 2 variant, H2AX, is phosphorylated during DNA damage. The hypothesis is that this phosphorylation opens up the chromosome in such a way that makes DNA repair more efficient. The other thing I want to mention is apoptosis. Again, this is also just an observation. H2B histone at serine 14 is phosphorylated during apoptosis. And to remind you, apoptosis is programmed cell death. Okay, next I want to talk about is histone ubiquitination. Usually what happens when we think of ubiquitination is that proteins, I'll just draw a generic one here, become hyper ubiquitinated in long chains using an enzyme called ubiquitin ligase. These proteins then are destroyed. These multi-ubiquitin molecules targeted to the proteasome, that structure in the cell that degrades proteins. That's not exactly what we're talking about here. We don't want to degrade the histones. At some point we do, because at some point we need to replace proteins. But in this case, ubiquitin is going to be doing something else. So what's different here is that H2A, these are its preferred targets, H2A and H2B receive a single ubiquitin. So they are mono-ubiquitinated. So what is it doing? Well, let's draw our piece of DNA here, and let's say this is a gene right here that we're transcribing, and the histones are still loosely attached to it, but enough is exposed so that the RNA polymerase can move along. What we think is happening here with ubiquitination is that this H2A or B is ubiquitinated, and that is necessary for the RNA polymerase to continue elongating the messenger RNA. So that step of elongation requires the ubiquitination of H2A or B. If it's not ubiquitinated, elongation would not proceed, or if it proceeded, it would not do so as efficiently as it needs to. Okay, next I'd like to spend a small bit of time, not too much time, but talking about summolation of histones. So what is a sumo group? Well, a sumo group is similar to a ubiquitin. It's different though because it has no role in protein degradation. The sumo group is added to lysines using a sumo ligase. Like ubiquitination, sumo also has a preference for H2A and H2B. Though it's still being investigated, typically when a lysine is sumolated, it decreases expression. How it decreases expression, we don't know. Some of the hypotheses is that it distorts chromatin structure, and another hypothesis is that it blocks transcription factor binding. Some evidence to support this, but not sufficient evidence to make any bold claims about what sumolation is doing. With the one exception that it does seem to be strongly linked to decreasing expression, but how it's doing it, the mechanism, is unclear. The last modification I want to talk about is histone ADP ribosylation. So what is ADP ribosylation? So let's once again draw our DNA, and we'll draw our histones on here. Now I'm going to draw this time something I haven't been drawing for a while, and that is H1. That links these histone octamers. And I'm drawing it this time because this is the histone that appears to be preferentially modified by an ADP ribose group. So ADP gets attached to H1. So it's appropriate we talk about this as our last modification because it's the first one and the only one we're going to talk about that doesn't modify the histone core here, the part that makes up the nucleosome. It's this linker one. So it's an interesting case. We know that when ADP ribose is added to the H1, that H1 is not displaced. It's still there. However, using electron microscopy, ADP ribosylation of H1 relaxes the chromosome, or relaxes the chromatin, I should say. Why it's doing that, we don't really know. It's rather a mystery. However, there is some evidence that this ADP ribosylation has a role in maintaining unmethylated CpG islands in promoters. So perhaps by loosening the association here, it's providing enzymes access here to make sure that these CpG islands remain unmethylated. Unmethylated CpG islands would promote gene expression. Though at this point, there's still a lot of speculation about what ADP ribosylation is doing. So another area that's wide open for future research. All right, well that ends this podcast. So I think it's time to have a short summary. We covered quite a bit in our discussion of histone modification. We talked about several kinds of modifications. We talked about acetylation, methylation, phosphorylation, ubiquitination, sumolation, and finally ADP ribosylation. There's other kinds of modifications as well, but I just talked about these 6. And we also discussed how these varied modifications of the histones leads to this histone code hypothesis. One that we don't really understand as well as we'd like to know. But we know different patterns of acetylation, methylation, phosphorylation, ubiquitination, sumolation, and ribosylation tells the cell specifically to turn on some genes and to turn off other genes. We understand the genetic code. We know how to read a gene and tell which proteins are being expressed. But we don't yet know how to read this histone code. We're learning some of it, but it's a slow process. But ultimately, if we can understand this histone code, it's really going to help us understand the epigenetic code. Okay, that's all I have for this podcast. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.